Hello, and welcome to Iman Phillips Biolab World. Philip is right now preoccupied, and I am turning a pipette, so it's taking a while.
Okay, I just filled it. You need to flick it.
Put it under some hot water.
Right now, I'm getting Iman to flick the tube.
Okay, so while I'm flicking this tube, Phil, why don't you talk about the experiment?
So today, our experiment is that we are trying to change the phenotype of E. coli by introducing new bacterial DNA into it
From plasmids.
Now,
Well, today's lab is about bacterial transformation, where normally a genetic material is passed from parent to offspring. Through mitosis or meiosis, or in bacteria's case, binary fission. But bacteria, some bacteria, have the capability of taking in DNA in the form of plasmids, which are small non-essential genes, really, but they do help the organism live. They're just small rings of DNA that code for certain proteins that aid the organism. And when a dead bacteria lyses, these plasmids are released into whatever medium they exist in. And some bacteria have the capability to take in another bacteria's plasmid, thereby altering their own genetic makeup by bringing in new DNA. So it's instead of going from parent to offspring, it's going from same generation to same generation. Okay, so what we're doing is we're setting up two solutions or two matrices, whatever you want to call them. We're setting up two of them. And one of them contains competent E. coli bacteria, and what that means is bacteria that have the capability to take in plasmids from another dead bacteria, and we're putting 50 milliliters, 50 microliters, sorry, of that E. coli cells into 5 microliters of plasmid containing solution, and we're doing 50 microliters into 5 microliters of sterile water. So the plasmids that we have contain the DNA for a gene to resist the antibiotic ampicillin. So if the E. coli takes in the plasmids, they will be resistant to the ampicillin.
Normally, E. coli is ampicillin sensitive, which are most diseases or viruses are bacteria that are sensitive to these, like ampicillin or penicillin. Ampicillin is part of the penicillin family. But some genes, they develop, like some bacteria, they develop this gene that allows them to be resistant to the penicillin or the ampicillin in this case. And what we're doing is introducing that plasmid that has that gene into the E. coli to try to make it resistant.
So our control for this experiment is a negative control where we have the water and the E. coli. Now that E. coli will be tested with the ampicillin too, and since it's going to be sensitive, it will die. If the E. coli that we put into the plasmid solution lives, that means that they took in the plasmid DNA, because it's the same E. coli, just one got put into a solution of water, one got put into a solution of plasmids. Okay, lift it up. Okay, so what we're doing here is I've got the bacterial solutions, and we are spreading them out on our agar plates, which is our medium for our bacteria to grow. So I've got the solution, taking a cotton swab, just making sure I got all the cells that I can get, and swabbing it onto the agarose, which is the medium for it to grow.
Just take pictures still as bad. No, they were videos.
Today's joke of the day comes in the form of a limerick. A bacterium named Bill and his brother went out for a drink with each other. In the midst of their clapping, they split their sides laughing, and each of them now is a mother.
Hi everyone,
Welcome to
Phil,
and Emod's
Biolab World.
Alright, so now we're back with our Bacterial Transformation lab
3 weeks ago.
3 weeks ago. So we've had our bacteria growing for that long. Or should say, dying. Okay, so right here we got
A negative control.
yes. No, it was 3.
We filled this with the
This is the
E.
water.
coli, right? Yeah, we used water.
This is the water test where there was no plasmid in the
E.
mix,
coli sample, yeah.
and
This is the plasmid mix. Look at all those bacteria. Yeah, those
so you see the
small white spots are the colonies that grew over the 3-week period.
and that's the plasmid containing one.
So it shows that our plasmid did allow the E. coli cells to become ampicillin resistant. Hence the growing.
Those are our results.
Yeah.
We'll see you next time on Emod and Phillips
Bio
Lab.
World.
Duh.